Good morning, students. Today we'll be having a short session on archaebacteria, in which we are going to discuss about the mechanism of halophiles. Okay, so let us start with our lecture today. So let us start with our lecture today. Okay, as you all know, halophiles are basically those bacteria which basically needs salts, okay, which needs salts for their growth. And one of the best studied halophile is a halobacterium. Is an halobacterium which consists of several species of the archaea with an aerobic metabolism, which requires an environment with a high concentration of salt, and many of their proteins will not function in low salt environments. Okay, it will not function in the low salt environments. Okay, so they grow on amino acids in their aerobic conditions, and their cell walls are quite different from those of bacteria. Okay, and ordinary lipoprotein membranes fail in the high concentrations, okay, of the salt. As we have discussed in the semester 3, I hope you remember, the membranes of the halophiles are basically different from that of the other bacteria. Why do they need different types of membrane? Because they grow in a very high salt concentrations. Okay, so they may be rod shaped or cocci shaped, or in color it may be red, purple, etc. And they reproduce using a binary fission by constriction and are motile. And halobacterium grows best at around 42 degrees Celsius in the environment. They grow best at 42 degrees Celsius in the environment. Okay, so I have taken up some of the photographs of the halophilic bacteria from the various sources available online, as well as some of them from the books. Okay, this one is a sample taken from a salt tun, okay, in Australia, viewed by a fluorescence microscopy. The range of cell shapes include cocci, rods, and even cubes. Now, okay, cocci, okay, some of them are even rod in shape. And these halophiles basically can be also isolated in our Gujarat region, where the salt tuns are located at various places in the Saurashtra. Okay, if you go towards Miana, okay, so near the Surajbari bridge at Little Inn of Kutch, there are very large salt tuns located. Almost in that area, almost around 20% of the salt which is being produced comes from the Little Inn of Kutch in India. Okay, in India, if you want to isolate halophile, near Surajbari bridge, Little Inn of Kutch is one of the best option, and another one is a Sambhar Lake in Rajasthan. And the halophilic archaea, the second photograph here, where you can clearly see the reddish type of coloration, okay, due to the pigments. A solar evaporation pond in extremely high in salt and mineral content, the archaea that dominate this hot saline habitat brilliant green pigments, okay. And the scanning electron micrographs of the Halobacterium salinarum, okay, now you can see the photographs here, scanning electron micrographs. Now these are the variety of colors, okay, you can clearly see in the photograph which are produced by the halophiles, and solitons changing color with the increasing salinity due to the various pigments different halophiles produced, and these pigments also facilitate UV resistance and or phototrophy. Okay, and or phototrophy, yes, some of the halophiles are also able to go for phototrophy also. You can see the variety of colors. Okay, these colors may vary according to the concentration of the salts which are present in these solitons. Okay, these are basically solitons here. And the UV resistance, these pigments facilitate UV resistance. Okay, as they are directly exposed to the sunlight, okay, throughout the day, okay, as well as due to the open spaces, there are chances they will receive high amount of UV radiation. So these pigments facilitate UV radiation. Now, like the cell wall proteins of the cytoplasmic membranes of the halobacterium are highly acidic, okay, are highly acidic, okay, but whereas in the case of regular bacteria which are not halophiles, this membrane is not highly acidic. This membrane is not highly acidic, but this acidity is due to the potassium ion, but not of the sodium ion. Okay, not of the sodium ion. This makes sense because potassium is the predominant cation in the cytoplasm of the cells of the halobacterium. In the cytoplasm of the cells of the halobacterium, okay, because its cytoplasm contains high amount of potassium. Besides having a high acidic amino acid composition, halobacterial cytoplasmic proteins typically contain lower levels of hydrophobic amino acids and a lysine, a positively charged amino acid, than proteins of the non-halophiles. Okay, so they will be having a high acidic amino acid composition. Okay, and have low levels of hydrophobic amino acids also. And it is also expected because of this highly ionic cytoplasm, more polar proteins would tend to remain in the solution, whereas less polar proteins would tend to cluster and perhaps lose its activity. This is how the mechanism works in the case of the halophiles. Okay, this is how the mechanism works in the case of a halophiles. The ribosomes of the halobacterium require high amount of KCl for their stability, whereas the ribosomes of non-halophiles do not require KCl. Okay, do not require KCl. So this is a main difference between ribosomes of the non-halophiles versus halophiles. And why do they need high KCl levels? The reason is simple, as this is an bacteria which can which can grow only under the high salt concentrations, which is basically required for their activity, and the environment in which they are growing will be also an alkaline as well as high salt containing environment. Okay, and extremely halophilic archaea are thus adapted in a highly ionic environment, and the cellular components exposed to the external environment require high amount of sodium for their stability, whereas the cytoplasmic components require high amount of potassium, okay, for their stability, with the exception of few extremely halophilic species of bacteria that also use KCl as a compatible solute. In no other group of prokaryotic cells do we find such unique requirements for such high amounts of the specific cations. Okay, these concentrations have been studied in the case of Halobacterium salinarum. Okay, this table given here just for your informational purposes. Okay, and to give you a better understanding of the halophilic cytoplasmic components. Okay, so iron you can clearly see sodium, okay, potassium, magnesium, and chloride ion. and chloride ion. And the concentration in the medium means in which they are growing. It may be salt water or salt medium in the laboratory, okay. And the concentration in the cells, okay, such as in the cytoplasm, cell membrane, etc., whatever it may be, okay. So why do they require sodium? Okay, because it maintains a high ionic environment, which is basically required for the acidity, for the highly acidic cell walls. Now clearly you can clearly see here the sodium contains in the medium is around 4 molar, okay, and the concentration in cells will be low, just 1.4, okay. Yes, it is greater than other non-halophilic bacteria, very high concentration, okay. It is greater than high other bacteria. Potassium contained in the medium is very low, 0.032 molars, but as I told you, the potassium content is very high in these cells, is around 4.6 molars, in the cells, okay. And magnesium ion is 0.13 molar, and in the cells is around 0.12 molar. Potassium ion required, okay, is around 4, similar to that of sodium, because it directly deals with the NaCl, okay. It directly deals with the NaCl in the medium, okay. So the concentration of chloride ion will be also same, but in contrast to the sodium, the chloride concentration in the cell will be high, okay, and will form KCl, okay, which is basically a soluble, solute also, okay. KCl, which is basically solute. Now the structure of the retinol bound to rhodopsin in species of the Halobacterium. This rhodopsin has the structure of Schiff base between the chromophore retinol and the lysine residue of a apoprotein, okay. And the Schiff base exports hydrogen with the light energy, as we have discussed in the photosynthesis. I hope you remember, in the case of bacterial photosynthesis, we have discussed halophiles, that it contains bacteriorhodopsin, okay. And in the bacteriorhodopsin, in this structure, it has a Schiff base, okay. What is Schiff base? Okay, Schiff base basically is an compound which contains structure similar to, okay, I am writing the structure, R1, R2, C, double bond, N, R3, okay. This one is a Schiff base, okay. This one is a Schiff base, and they are considered as a subclass of, I think, imines, and they are also found uh, with just various other structures also, okay. So basically the Schiff reaction, okay, usually occurs with the basic conditions of aromatic amines to form a Schiff base, okay. And these bases are basically used as catalysts or pigments, dyes, intermediates also, okay. So they have a variety of function. Now this Schiff base here, okay, which is basically a double bond here, which is shown in a trans form as well as the cis form, and they are involved in the export of hydrogen ions, okay, in the presence of the light, okay. Here hydrogen is added here, and the double bond rotation which occurs in the transform, and this one is a structure basically of a Rhodop spin. Okay. Now the model for the mechanism of bacterial rhodospin. Okay. Now RETC, which is a retinal one, C form means cis form, T form means transform. Okay. The light which will be absorbed is basically 570 nanometer, okay. This is basically being studied in the case of hello microbiome. Okay, sorry, hello bacterium. Okay, and light of 570 nanometer converts protonated retinal of bacterial rhodospin from trans to the cis form along with the translocation of protons. Now this hydrogen basically moves this word, okay, proton to the outer surface of the cytoplasmic membrane and thus establishing a proton motive force. On this side, proton motive force will be generated, and finally with the proton motive force ATP will be generated with the help of the enzyme ATPase. Okay, I am repeating the process again. In the presence of light at 570 nanometer, which converts the protonated retinal bacterial rhodospin from the transform to the cis form, okay, along with the translocation of hydrogen to the outer surface of the cytoplasmic membrane, which basically helps to establish a proton motive force, and which with the help of the ATPase enzyme and the force generated, ATP will be generated. Okay. Now moving forward with the further same, if you want to study whichever diagram you want to use, you can use, you are free, it's purely depend on you. Okay, the chemiosmotic coupling between the sun energy bacteriospin and phosphorylation by ATP synthase during the photosynthesis in Halobacterium salinarum. Okay, in the Halobacterium salinarum. And the Halobacterium species can be found in the Great Lakes, Dead Seas, Lake Magadi, okay, and any other waters with high salt concentration. In India, Sambar Lake and near the Little Rhine of Kutch are the best sources where we can isolate halophiles, and the purple Halobacterium species owe their color to the bacterial rhodospin, a light sensitive protein which provides a chemical energy for the cell by using the sunlight to pump the protons out of the cell. The resulting proton gradient across the cell membrane is used to drive the synthesis of the energy carrier ATP, and thus the protons flow back in and they are used in the synthesis of the ATP. Okay, for example, this proton flow can be emulated with a decrease in the pH outside the cell and causing a flow of hydrogen ions. And the bacteriospin protein is chemically similar to that of light detecting pigment rhodospin found in the vertebrate retina. Okay, let's have a look of some of the photographs, okay, of the halophiles. Okay, this one is a photograph from Great Salt Lake, Utah, a hypersaline environment in which the ratio of ion is similar to that in the seawater, but which concentrations of ions are several times that of sea water. And you may see slight greenish coloration, which is basically from cyanobacteria and green algae, which is basically from cyanobacteria and green algae. Okay. Now next one photograph is basically a aerial view near San Francisco Bay, California, of a series of seawater evaporating ponds where solar salt is basically prepared, and the red purple color is predominantly due to the bacteriorubberins and bacteriorubspins in the cells of Haloarchaea, in the cells of the Haloarchaea. Okay. Now this one here is photograph is from a Lake Hamara, Wadi Il Natron, Egypt. Okay, which is basically a bloom of a pigmented haloalkalophiles which grow at a pH of about 10 in a soda lake. Okay, and you can see the deposits here. Okay, deposits which are basically deposits of the trona. Okay, the formula is given here. Okay, you can see it's basically in carbonates. It is basically in carbonate. Okay. Now scanning electron micrograph of the halophilic bacteria including a square archaea which is present in the Salton of the Spain. Okay, not just one species, there are various other species. Okay, where the square shaped ones are basically archaea. Okay, and there are also other bacteria also which are basically halophilic, Okay,